So roles of monosaccharides: one, they are a source of energy because they have large number of carbon-hydrogen bonds. The second role of monosaccharide is that they are the building blocks for larger molecules, such as polysaccharides, like glucose is used to make starch. So glucose is used to make starch and cellulose also. I'll talk about this later on, but yeah, two roles of monosaccharides. One, they act as source of energy. Two, they are the building blocks for larger molecules. And remember, monosaccharides are monomers, right? So this was monosaccharides, and moving on to disaccharides. Disaccharides, right? Di basically means two. So when two monosaccharides combine together, they give a disaccharide. And when two monosaccharides they combine together, the bond that is formed is called a glycosidic bond. So glycosidic bond is a covalent bond which is formed between two monosaccharides by a condensation reaction. What is a condensation reaction? Condensation reaction basically is that you're releasing water in the process. When you combine these together, you make a glycosidic bond, and this would release water. So there are three different disaccharides you need to know. The maltose, which is made up of, again, it's a disaccharide, so two monosaccharides combined together. The first one is alpha glucose, and the second one is also an alpha glucose. And the roles of maltose is that it acts as the energy source in germinating seeds. Now, glucose, right? There is two types: the alpha glucose and the beta glucose. And I'll explain both later on. For now, you only need to know there are two types. Alpha glucose makes up maltose. So two alpha glucose combine together to make maltose. Sucrose is when alpha glucose and fructose combine together. And the role of sucrose is that it transports substances in the phloem. Again, I'll explain what it would transport. For now, they transport different kinds of sugars, basically the compounds that make energy, right? That releases energy. Sucrose and sugar is basically transported in the phloem. And what is lactose? Lactose is the energy source that is found in milk. People say that they are lactose intolerant. They can't handle lactose, so they can't drink milk because lactose is found in milk. And lactose is made up of alpha glucose and galactose. Glactose, galactose. Right. So three kinds of disaccharides. Remember their roles and remember the monosaccharides that they are made up of. So finally comes polysaccharides. Polysaccharides are long chain of monosaccharides added together by a glycosidic bonds. So not two monosaccharides, but many monosaccharides combined together to make polysaccharides. So thousands of monosaccharides join to form a macromolecule. Polysaccharides are not sugars. Most important polysaccharide is starch, glycogen, and cellulose. So starch is found in plants, and glycogen is obviously found in humans, and cellulose is found in plants as well. So glycogen and starch is formed by condensation of alpha glucose, whereas cellulose is formed by the condensation of beta glucose. So glycogen and starch, the monosaccharides that make it up is the alpha glucose, whereas cellulose is formed by beta glucose. And both of these reactions are condensation reactions. Excuse me, but why are polysaccharides not sugars? Polysaccharides are not sugars. So sugars, right? Sugars can be one. They have sugars have two main roles. Sugars can be broken down, and the second main feature of a sugar is that it can act as a building block, right? So basically, glucose acts as a building block. Again, if you have one or two sugars, they can combine together. And when the product is formed, right, that is not a sugar because basically polysaccharides are many sugars combined together. So yeah. So moving on to starch and glycogen. Starch is a mixture of amylose and amylopectin. And amylase, amylose, and amylopectin, right? Amylopectin. Yeah, these two are very important. I've seen like 4 or 5 questions from the recent years. So amylase is made from alpha glucose of several 1,4 links. So remember, starch is made up of these two things, amylose and amylopectin. Amylose, amylase, right? Amylase is made from alpha glucose and which have several 1,4 links. 1,4 links are basically the bonds formed between amylase, whereas amylopectin is made from alpha glucose of several 1,4 links and 1,6 links. 1,6 links plus 1,4 links. So if a question asks you to identify if the molecule is amylopectin or amylase, if it's amylase, then it will only have 1,4 links. The bonds will only have 1,4 links. But in amylopectin, there would be 1,4 plus 1,6 links. So that's the difference. And there is another difference. Amylase, right? Amylase is coiled, thus it's very compact. That means it can store large amount of energy in small spaces. So a feature of amylase is that it is very compact. It can coil together. Due to the presence of many side branches, amylopectin is rapidly digested by enzymes. This energy release is quick. So another a feature of amylopectin is that it has side branches. Side branches are basically CH3, CH2, CH3. You don't need to know the names of the side branches. What you need to know is that amylopectin has side branches. That's one of the features. The second feature is that it has 1,4 plus 1,6 links. And amylase, right? The first feature is that it's compact. And the second feature is that it has 1,4 links only. 1,4 link only. So moving on, right? Starch is only found in plant cells. Very famous. This is very common. And animal cells have a similar molecule called glycogen. Both of these, right? Both the starch and glycogen act as energy storage molecules. So glucose, right, is soluble in water, which can change the water concentration of cells. This is why it is stored in polysaccharides. So glucose, remember, it's a monosaccharide. Glucose is a monosaccharide, and we can't simply store glucose in our body because of many reasons. One of them is that it is soluble. So when it dissolves in water, right, it can change the concentration of water. What this does is that because the concentration of water is changing, the ion concentration would also change. So it would either increase and decrease. Because of this, ions will move randomly in and out of cell, and this can cause many problems. So that is why it's not glucose is not used as glucose. We can break glucose into ATP. So glucose is converted to ATP. That is the reason why we use ATP instead of glucose. So glycogen, it has similarities to amylopectin. So glycogen is very similar to amylopectin because one, it has 1,4 plus 1,6 links. That's one of the reasons. It has 1,4 and 1,6 glycosidic bonds or links. And the second reason that it's similar to amylopectin is that glycogen has more branches compared to amylopectin. So glycogen is also a branched, it's a branched compound, and it has more branches compared to amylopectin. So glycogen is branched. It has 1,4 and 1,6 links, but it's more branched compared to amylopectin. So yeah, so Glycogen, remember again, it's found in animal cells, whereas starch, again, it's found in plant cells. So another polysaccharide is cellulose. So cellulose, glycogen, and starch, all of them are polysaccharides or macromolecules. So cellulose is found in cell walls of plant cells, and it's a polymer of beta glucose. Remember, glycogen and starch were made up of alpha glucose, whereas cellulose is made up of beta glucose. Unlike glycogen and starch, it is made up of beta glucose. So cellulose makes up about 20 to 40% of cell walls. This is a fact you need to know. And other than that, cellulose fibers are freely permeable and allow water solutes to enter and leave the cell wall. Note that cell membrane is different from cell wall. It is not freely permeable, rather it is partially permeable. So cell membrane is only partially permeable, whereas cell wall is fully permeable. So cellulose are unbranched, so they are close to each other. Hydrogen bonds can form to make cellulose strong. And every second beta glucose is flipped. I don't think you need to know this because this fact is for diagrams, but you won't find diagrams in the exam really. So this is pretty much cellulose. So that was carbohydrates. So now moving on to lipids. Lipids are organic molecules which are insoluble in water. What is lipids made up of? The monomers that make up lipid are fatty acids and alcohols. So fatty acids are monomers, and they are long chains of hydrocarbon. Some acids have single bonds only. That means they are saturated fatty acids, whereas some fatty acids have double bonds. That means they are unsaturated fatty acids. So when a fatty acid or a carboxylic acid combines with alcohol, in this case, the alcohol is glycerol, what you get is a triglyceride. What is a triglyceride? Triglyceride are basically three fatty acids combining with one glycerol molecule. And why are three fatty acids used? Because remember that 1 mole of glycerol has 3 OH groups. 3 OH groups are found in glycerol, and only 1 OH group is found in fatty acid. So one molecule of fatty acid has 1 molecule of OH, but glycerol has 3 OH molecules. So when 3 of these fatty acids combine, these bonds will form. The OHs will combine together and they would release water. Are lipids absorbed as triglycerides in the body? Someone is asking a question. Are lipids absorbed as triglyceride in the body? I don't understand your question. Are lipids absorbed as triglycerides in the body? Lipids are not absorbed by the body. It's stored by the body. You'll store lipids and they're not really used as energy sources. I'll get to triglycerides, the roles of triglycerides. Let me finish this and I'll get to triglycerides first. So triglycerides, right? Again, it's made up of 3 molecules of fatty acid and 1 molecule of glycerol. And triglycerides are insoluble in water. They are soluble in organic solvents like ethanol. So what is the role of triglycerides? They act as energy reserves because they will yield more energy because they have carbohydrates due to more carbon-hydrogen bonds. So they have a lot of carbon-hydrogen bonds. That is the reason triglycerides will release a lot of energy. And another reason is that triglycerides would insulate your body from heat loss. They can absorb heat and act as insulators. And they're also a metabolic source for water. When triglycerides react with oxygen, it will give water. So remember this, that triglyceride plus oxygen would release water. And this reaction is known as condensation because water is being released. So 3 roles of triglycerides. 1, they are good energy reserves. 2, they insulate our body, and 3, they would release water. So it's acting as a source of water as well. Someone had a question. Are lipids absorbed as triglycerides in our body? So lipids, right? Lipids there are many forms of lipids. You will have phospholipids, you'll have triglycerides as well. So lipids are not formed only stored in the body as triglycerides. You can form them as phospholipids as well. These two are the main types of lipids you need to know, the phospholipids and triglyceride. I think there is one more lipid that's called it's found in the cell membrane. Yeah, and there is steroids as well, but there is another. So there is multiple lipids that can be formed. So there is one more. It's found in the cell membrane. I forgot the name, but you can check out my last class. You'll find the phospholipid. Yeah, cholesterol and one more. So there is multiple ways lipids can be stored in our body. That's the main point. Yeah. So again, phospholipids. Moving on to phospholipids. What is phospholipid? I talked this in the last class about cell membranes. It's found in the cell membrane, and one end is soluble in water. The heads, right, are soluble in water, whereas the tails are insoluble in water. So the two tails are hydrophobic. They don't like water. They don't form bonds with water, whereas the heads are hydrophilic. They love to form bonds with water. And one of the 3 fatty acids molecule is replaced by a phosphate group. So the difference between triglyceride and phospholipid is that in triglycerides, you have 3 fatty acids, whereas in phospholipids, you have 2 fatty acids plus 1 phosphate group. Yeah, this is very simple. It's very basic. I talked about this in big detail in the cell membrane topic. Go check that out. So yeah. And finally, proteins. Proteins is a very important topic. So all proteins are not enzymes, but all enzymes are made up of proteins. This is a random fact. Don't focus on this too much. So yeah, so some hormones such as insulin and glycogen are proteins. And proteins are made up of amino acids. The building block or the monomers for proteins are amino acids. And an amino acid, right, has amine group, NH2 basically, on the left side, and carboxylic group or CO2H on the right side. So at the very right end, you'll have a carboxylic group. At the very left, you'll have the amine group. And you'll have one H and a side chain. This side chain will tell you what type of amino acid you have. And remember, the simplest amino acid that can be formed is glycine. Glycine is a carbon bonded to 2 hydrogens and one carboxylic group and one NH2 group. So you'll have NH2 and carboxylic group in all amino acids. But the thing that tells you which amino acid that is the side chains. So remember that proteins, when the type of bonds in proteins are known as the peptide bonds. So proteins form peptide bonds, and what type of bonds do carbohydrates form? Carbohydrates, they form glycosidic bonds. Proteins form peptide bonds. Yeah. So in a peptide bond, right, one OH is lost from the carboxylic group. Remember, carboxylic is CO2H. So one hydrogen and one oxygen from the carboxylic group and one hydrogen from the amine group. So when these combine together, one OH plus H, you get H2O. You are releasing water. Therefore, a peptide bond, when it's formed, it's a condensation reaction. So two peptides, they form together to make a dipeptide. And many peptides, they combine together to form polypeptides. So when you have a lot of peptide bonds, that will be called a polypeptide. And peptide bonds are covalent bonds, like glycosidic bonds. And peptide bond is stronger than tertiary structure bonds. I'll get to tertiary structures. So yeah, that's basically peptide bonds. So you also have to know the structures of proteins. Proteins, right, have 4 types of structure. The primary structure, which is the sequence of amino acids that make up the polypeptide chain. So basically, the number plus the sequence of amino acids. That's the primary structure. So it is the order and number of amino acids. Yeah, basically the same thing, number and sequence of amino acids. Whereas the secondary structure tells the shape of the amino acid chain. The secondary structure can either be alpha helix or beta pleated sheets. 2 types of secondary structure, alpha helix or beta pleated sheet. And alpha helix, right, they are formed due to hydrogen bonds forming between oxygen of the COOH. So oxygen from the carboxylic group and the hydrogen from the NH2 group. So when these two, the hydrogen from the NH2 and oxygen from the carboxylic group, they combine together, they form a bond. That bond is formed and you form a alpha helix. And sometimes hydrogen bonds would be looser. It would be much loose compared to the alpha helix. So if the hydrogen bonds are loose, then you will get a beta pleated sheet. In alpha helix, you have strong hydrogen bonds compared to beta pleated sheets. Yeah. So hydrogen bonds are easily broken by high temperatures and pH changes. So secondary structure, right, has 2 types, the alpha helix and the beta pleated sheets. And these two are easily broken by 2 things. So if you have a change in temperature and a change in pH, both of these can change the structure of secondary structures. Right. What is a tertiary structure? It is the 3D shape of proteins. It can be either globular or fibrous. And tertiary structures have 4 different types of bonds. The hydrogen bonds, which form between strong polar groups like NH or CO from the carboxylic group and OH. And these can be broken by pH and temperature changes. So disulfide bonds are the second types of bond that is found between tertiary structures. And disulfide bonds are basically 2 cysteine molecules which contain sulfur atoms, right? And this can be broken by reducing agents. And this is a covalent bond. So when 2 cysteine molecules, cysteine is basically an amino acid, like glycine, it's an amino acid. So when cysteines, right, they have sulfur in them. So when you have sulfurs can form bonds between each other, and that bond is a covalent bond. And this can be broken down by reducing agents. Disulfides can be broken by reducing agents. Hydrogen bonds can be broken by pH or temperature changes. And the third type of bond you will form is the ionic bonds. Ionic bonds are found between amino acids and carboxylic groups, and they can be broken by pH changes. Ionic bonds are simply the positive and negative charges that attract each other. So they can be broken down by pH changes. Yeah, so 4 types of bond. There is one more, the hydrophobic interactions that happen between R groups, which are, these types of bonds are non-polar. So the hydrophobic interactions between R groups are non-polar. Yeah, so 4 types of bonds, remember the names and the reasons that they can be broken. So what finally comes the quaternary structure. So I had the primary, the secondary, and the tertiary. Now comes the quaternary structure. Quaternary structure are made up of 2 or more polypeptide chains. Quaternary structure are the structure that makes up 2 or more polypeptide chains. So when two or more chains, they interact together, right? They are called the quaternary structure. So tertiary structures is formed by different R groups binding to each other. Yeah, that's another fact. And finally, hydrogen bonds form between oxygen and CO groups. I told you earlier. Yeah, so that was the types of structure you need to know. And remember that there are two types of proteins, the globular proteins and the fibrous proteins. Globular proteins, right? These proteins are molecules that curl up. So globular, curled up, they can be in the shape of balls or different types of shapes. But fibrous proteins, from the name, they are long fibers or long strands. Examples of globular proteins are enzymes, and examples of fibrous proteins are collagen or keratin. And fibrous proteins are not soluble in water. They mainly have structural roles, whereas globular proteins are generally soluble in water and they have metabolic roles. For example, enzymes, they decrease the rate of reaction. Is insulin globular protein as well? Yeah, it's a insulin, right? Insulin is uh, having is involved in metabolic roles. And if something is fibrous, it will mainly have structural roles. But insulin does not have any structural roles. What do I mean by structural roles? Like keratin, right? They form hairs, nails, and outer layer of the skin. But I think insulin is globular. I'm not sure. You can check it out. But insulin has a metabolic role, so I'm assuming it's globular. I'm not sure though. I can be wrong. It is, yeah. So it is. Someone is saying it is a globular protein. So yeah. So I want to move on to globular protein examples. Example of globular protein, a very important one, is hemoglobin. Hemoglobin carries oxygen, which is found in red blood cells, and it has 4 polypeptide chains. Hemoglobin has 4 polypeptides chain, so it is a quaternary structure. Basically, any globular protein will have the 4 structures: the primary, secondary, tertiary, and quaternary structures. And each polypeptide chain is called a globin. So remember, 4 polypeptide chains, each chain is called a globin. And hemoglobin is made up of 2 alpha globin chains and 2 beta globin chains. And remember that each chain holds a heme group. And each heme group is attached to one iron atom. And each iron atom can form bonds with one oxygen molecule. So in one red blood cell, you can, since there is 4 chains, so there would be 4 heme group. So there would be 4 iron atoms, and therefore there would be 4 oxygens in one red blood cell. So pretty basic. And yeah, another example of then we have fibrous proteins. Example is collagen. And most common protein that is found in animal cells, it's the collagen. And it has 3 polypeptide chains which wind around each other to form a triple helix. And these 3 chains, right, are held together by hydrogen and covalent bonds. And what is the advantage of collagen? You also need to know the advantages. It's very flexible and it has high tensile strength. So it can withstand pressure. That is the reason it's found in skin. Collagen fibers, according to they can stand the force according to the force they withstand. And every third amino acid is a glycine molecule in a collagen fiber. And collagen fibers form cross links and are staggered to increase the strength. So the reason they can withstand high pressure is that collagen fibers have cross links and are staggered. Staggered. This is a very specific word. Staggered to increase strength. Someone is writing something. It's a hormone, a peptide hormone though. Yes, hormones, uh, all hormones are not proteins. There are, but all proteins, right? They will have peptide bonds. There are steroid and protein hormones. Peptide hormones are made from small amino acid chains. Yeah. So that was kind of like small globular protein. So that was basically the compounds that you need to know for the IMAT. I want to finish enzymes as well. How much time do I have left? I think we can finish enzymes. So what is an enzyme? Enzymes, remember, are globular proteins that increase the rate of reaction by lowering the activation energy. And there is two types of the enzyme theory. That the first one, which is the most accepted, is the lock and key. Lock and key, it's when the shape of the active site does not change and it's complementary to the substrate. So for example, this is the enzyme. This is a very bad enzyme. This will be very complementary to the substrate. So the substrate will lock into the active site and the reaction would happen. Induced fit is when the tertiary structure of enzyme changes as the substrate approaches the enzyme. So in induced fit, the substrate, the active site would change shape according to the substrate and it would fit in. So that's why it's called induced fit. Now, enzymes, right? They operate inside a cell. So the enzymes that operate inside cell are called intracellular. The ones which work outside the cell are called extracellular. Yeah, so this is what I was talking about, the induced, the lock and key mechanism. And I told you what induced fit was. And yeah, so I don't think you need to know a lot about this, but the factors you need to know, the factors which affect the rate of enzyme activity. So there is 5 different factors. The first one is the substrate concentration. What this basically means is that that the substrate concentration, right? As the substrate increases, as the substrate increases, the enzyme activity would decrease. Enzyme activity decreases because remember that there is only a limited amount of enzymes. And if you have a lot of substrate, right, that can't be filled up by all of the enzymes. So if, for example, there is 10 enzymes, but you have 5 enzymes, 5 substrate, but you want to increase the substrate level. So let's say from 5 you make it 20 substrates. You can't fill up all of the 20 substrate with the enzyme because you only have 10 enzymes. So this would decrease the enzyme activity. So that was substrate concentration. What is enzyme concentration? Enzyme concentration is that if you have high concentration of enzyme, then this means that not enough enzymes, enough substrate is present to bind to all the active sites. So the rate of reaction basically stops. So if you increase the enzyme